This video will give you an overview of the bulk RNA-seq workflow on BWB. The workflow starts at the Start widget. This widget specifies the majority of the inputs and outputs for executing this workflow. At the top, you see the work directory, where this directory specifies the majority of the outputs are going to be stored. Below that are directories to store the genome and transcriptome related files that are downloaded or generated from the workflow. You can store generate files from the genome in the genome folder, download files in the downloads folder, and this includes the URLs for the downloaded files. The download URLs that are currently loaded are reference files. This includes the Ensemble GRCH38 reference genome, release number 110, and the GTF annotation file, GenCode version 44. Below that are locations to store trimmed and aligned files in different directories. And then there's a section to find BAM files in the aligned files directory. And here, there's a star version to use for the workflow. And then there are other entries that are related to AWS S3. Now, to the right of the Start widget, there are a couple of widgets related to reference file download and generation. In the Download widget, this is where the FASTQ reference genome and annotation GTF file will be downloaded. The remaining three widgets are used to format the reference genome to 10x genomics format, and then create a transcriptome and star index. The next set of widgets are responsible for processing FASTQ files. If you are getting files from S3, first download the files from S3 with this widget. Then, it will filter the S3 files for only FASTQ files in the directory, so that these FASTQ files are selected to be trimmed in the next step. In the next step, Trim Galore is responsible for trimming the FASTQ files. And the final widget in this row will filter just for the trimmed FASTQ files. The next row of widgets is responsible for alignment and quantification. Taking the trimmed FASTQ files from the previous step, you can use star align to do the alignment of your samples to the reference genome. The current configuration of the widget allows paired end files as long as the paired end files are named the same, but have altering file names for the FASTQ pair. For example, R1 for one file, followed by R2 in the other. To disable paired end files, disable this entry, alternating paired end files. After running star align, you'll get a bunch of output files. Most importantly, the align BAM files and star's version of gene expression count files. In the next step, Salmon is used to quantify gene expression from the BAM files, and it uses the transcriptome and annotation files generated earlier. As outputs, each BAM file will have their gene and transcript quant files generated separately. The last step is to combine the quant files obtained from Salmon for all samples and create a tidy spreadsheet with all genes and transcript quantification listed for all the samples. At the bottom, there are two optional steps you can do. If you want to store outputs to S3, the S3 upload widget can upload those outputs to the specified S3 bucket. And in the other widget, if you want to filter Y reads from the aligned BAM files from star align, you can do so with the filter Y reads widget. In the widget's entries, you will need to set the location of the aligned BAM file directory from star align, the output directory to store the filtered reads, and optionally provide the directory with the FASTQ files for the samples. If you provide FASTQ directory, the widget can split their FASTQ files into two sets, one with just the Y chromosome reads, and the other without the Y chromosome reads. With all the workflow processing steps covered, here's how you can modify the workflow if you do not want to use the S3 widgets, or if you have your own set of FASTQ files outside of S3 that you want to run in the workflow. First, delete the S3 download and S3 upload widgets. Then, open Filter FASTQ files and specify the directory with your FASTQ files. When you start the workflow from the Start widget, the current configuration does not make the Filter FASTQ widget run automatically. So you need to run the widget manually by clicking on this blue Start button. Or if you want to automatically start the widget from the beginning, you can connect the widget from the Start widget to the Filter FASTQ widget. Select any input to go to the output's trigger, then set the run mode to triggered. Finally, select trigger. This allows the Filter FASTQ step to run after the Start widget is finished running. That concludes this video. Hope this video provides you with enough information to run this workflow.